Today's topic for discussion is methods for herbarium preparation. Preparation of herbarium involves a series of operations. Let us discuss all the activities required for the preparation of herbarium. Number 1 is collection. We have to collect plants. Collection should be carried out on a clear warm day so that the plants dry rapidly. The plants should be collected if possible with flowers and fruits. The undamaged specimens should be collected with a great care. The specimens should be collected from different localities. Whenever possible, all the parts of the plant should be preserved along with the roots. Second step in preparation of herbarium is pressing. After collection of the plant, we have to press the plant. It is also one of the important technique. Pressing is one of the technique. The collected specimens should be carefully displayed by placing them in a center on the pressing sheet. Pressing sheet or blotting papers of rough quality or old newspaper we can use. If specimens are large enough, they should be bent by giving them shape of V, N or W extra branch. In this way we can press. Extra branches and leaves can be removed to avoid overlapping of the plant parts. The specimen should be placed for pressing in such a way that the bundles are uniform in thickness in the middle and on the sides. The pressing sheets loaded with the specimen should be kept one above the other horizontally and then tightly bound in a plant press to some extent a uniform pressure all over the sheet should be there. This is a pressing technique after collection of the plants. Next important technique, that is drying and poisoning. For effective drying of the plants, the drying paper should be replaced by fresh ones every day. The changing of drying papers is repeated every day for about fortnight or until the plant specimen appears perfectly dried. In the humid climate, the changing of the papers is done twice a day to have good results. If the wet papers are not replaced by fresh dry papers, the specimens get infected by several types of fungi and bacteria, and that specimen, that plant material get spoiled or that get deteriorated. Drying in intense sunlight causes plant specimens to lose their original color and becomes black. Artificial heat may be given if the weather is too humid. The dried specimen is then poisoned, usually with the saturated solution of mercury chloride, that is HgCl2 in alcohol, or we can use a spirit. The lauryl pentachloride is a safer substitute of mercury chloride. The specimen is either dipped in the solution by holding it with tongue and apply the solution by means of a brush. Care should be taken that the solution does not come in contact with the skin. After some time, when the alcohol evaporates and specimen becomes dry, it is mounted on herbarium sheet and labeled. Next important step in preparation of herbarium is mounting and stitching. After drying and poisoning, the plant specimen is mounted on a herbarium sheet which is made up of thick, handmade, durable card sheets having standard size of 29 cm into 42 cm. The dried specimens are glued on herbarium sheets and the stem or branches and petioles of the leaves are stitched. Stripe of brown gummed paper or adhesive coated cellophane tapes may be used instead of stitching with threads. It is advisable to mount one specimen per herbarium sheet. Dissected and loose parts such as flowers, fruits and seeds are kept in a paper packets and pasted to the mounted sheet. Next, another step which is an important step in preparation of herbarium is labeling. After mounting the specimen on the sheet, a label is glued on the lower right hand corner of the sheet to provide information about the plant gathered during the field visits, which has collected from field visit. The format and size of the label, which is sometimes printed blank for the information on the paper. That sheet is known as herbarium sheet, and that is to be filled and is about 4.5 centimeter to 7.5 centimeter space. The label should be furnished. The following information should be added in that table. We have to write name of the organization with which specimen plant originated, name of the family of which specimen plant belongs, botanical name of the plant with authority, locality of collection including place, the date of collection we have to write, the habit of the plant specimen, that also we have to add, field note and collection number, name of collector, vernacular name of that collected plant and local use of plant. All these information we have to write on the right side of the herbarium sheet, on which a table is given for information. Another last step in herbarium preparation is filling and storing. Plant specimens which have been properly mounted and identified are filled systematically in a special wooden or steel cabinets with pigeon holes allotted with different families. The herbarium sheets loaded with specimens are filled inside the folders, which are of various colors schemes indicating the species, genus, family, geographical area, etc. In most of the Indian herbaria, as also in Kew, the specimen plants are arranged and stored according to Bentham and Hooker system of classification, although Angler and Prandtl system is also recommended. The permanent collection of herbarium specimens requires its proper protection and care against damages by fungi and insects. A periodical fumigation with chemicals and repoisoning by brushing with solution of mercury chloride or lauryl pentachlorophenate and using insect repellents such as dichloro, diethyl trichloro or trichlorothane would be used to avoid the damage of collected plants. This is all about the methods for herbarium preparation. These are the techniques which are used for herbarium preparation.